polar cell polarity pathway may be involved in many other processes, in developmental processes, and so on and so forth. So the combinatorial was, can you still hear me? It seemed like something went strange.
Yes, I apologize. Okay. The recording hadn't started, but now you're good.
Okay. All right. Yeah, so the combinatorial is too large. Also, it made the ontology super complicated, and then there were all kinds of errors and inconsistencies, like the stuff connected in the wrong place in the ontology. And well, many of these terms have already been obsoleted. Yeah, so I don't know, this is a bit out of place, but just to say that. Oh yeah, no, sorry. I know what okay, sorry. So another approach to capture the complexity of the biology that we're trying to capture in papers is using annotation extension. So that's a second strategy. And so instead of pre-composing exactly the terms, we have been adding annotation extension to provide additional context. And this allows us to link activities to process components, as well as input substrate, and also terms from other ontologies like cell types from the cell ontology, anatomical structure from Uberon and all kinds of other anatomy ontologies. And so we have like a number of external ontologies that we link to. We also have substrates from Kebi, for example. So this is what something like this look like. You can have NET4 has a ubiquitin ligase activity, and that's part of negative regulation of transcription by RNA polymerase II, and that's part of the DNA damage response. So this is a post-composed term, right? So the composition of like the putting together of all the GO terms is made, in this annotation extension. So this we still use, but it's got its own limitation because first of all, it's a bit limited in scope because the annotations are not usually visible by any Go tool. Like we are not aware of any Go tools that use these things. And also it's not amenable to pathway-like representation, which was really something that we thought was missing. So this is just to illustrate why annotation extensions are different from pathways. This is the same annotation with the extension I just showed. And so, if like to have a pathway, you want to link together multiple activities. And so here you can say, that the ubiquitin ligase activity negatively regulates an RNA polymerase activity. So this is allowed in this data model. However, you cannot name which gene it affects. So you're not allowed to name that this is the activity of a certain gene, and that's in part also because of the nexting of the extensions and so on. But the data model is kind of too simple to allow this, precise assignment of a gene regulating an activity. So it helps provide some context, but it doesn't fully do the job. And so this is why we developed something called GO Causal Activity Models, or GO-CAMs. So they provide a system to extend Go annotations. So it checks the box of being able to provide context. So we can produce the component cell tissue where the function takes place. We can capture the substrates of molecular functions. We can capture a molecular function belongs to which process, and we can even nest them. So you can have like a signaling pathway, which is part of a cellular pathway or developmental pathway. And it also defines, and this is the big difference with the annotation extensions, it defines causal connections between activities. So you can describe how gene 1 affects, like the activity of gene 1 affects the activity of gene 2. And so it allows network representation and pathway visualization and analysis. So this was published already soon 7 years ago. And so this is the data model that we have in the paper and is still what we use, is the central point, the central part of the GoCam is a molecular activity. And then we have relations to all what we call context. So the molecular activity is enabled by, and these are all relations from the relations ontology. It's enabled by an active entity, meaning a protein or a gene or a complex. The molecular activity has an input, let's say a target or a substrate. The molecular activity occurs in a location, and this can also be nested. You can say occurs in the nucleus of a muscle cell, of a neuron or something like that. The molecular activity can be part of a process, and like I said, that can be nested too, and it can occur during a biological phase, for example, during development. And the big difference with the annotation extension is this is causally upstream of another molecular activity. So the causal relations from the relations ontology, they link molecular functions together or activities, and they can be either positive or negative. Like, we express the directionality of this. So this is a decision tree showing the different types of relationships we have. So we have either it's a regulation relation, a function regulates another function. It's either positive or negative, and then it's either direct or indirect. And so that leads to, you either directly positively regulates, indirectly positively, directly negatively, indirectly negatively. Also, you can have an activity which is just constitutively upstream of another activity. It's always there, there's no regulation. Or this is useful for metabolic pathways. An activity can provide an input, like I showed in the ergosterol example. The activity provides an input for the next activity. It doesn't regulate it, it's just its output is the input to the next activity. We have something else that we use not so often is removes input for. So far the examples we have for this are competing post-translational modifications. So there's some sites that if they are phosphorylated, they're not available for ubiquitination or they're not available for methylation. This happens a lot in the histone code, for example. And so we capture the relation like this because it's not exactly a regulation, because a regulation, we define it as the protein acts on the other protein. Here it acts via the input. So it's making the input not available. And then we also have undetermined. So sometimes we don't know how two activities are linked together. So we can say that one activity is causally upstream of another activity, either with a positive or a negative effect. And so this is the GoCam model view for the regulation of the RNA polymerase activity by NET4. So NET4 has ubiquitin protein transferase activity, has input PAL2. It's part of ubiquitin-dependent protein catabolic process, part of the DNA damage response. This happens in the nucleus. And this activity directly negatively regulates the RNA polymerase activity of PAL2. And so this one does transcription, and that means that this negatively regulates transcription. So this is what the network allows us to infer. And so, yeah, having these, the ability to do GoCam, it also really a bit shifts the way that you think about the biology, because it's a bit different from just capturing what you see as results in a paper. You're trying to really model with this, how the activities connect. And so this ended up motivating quite a few changes in the ontology. It's not just limited to that, but it's certainly a big reason. So we started to do a massive ontology refactoring in 2019 to also address the issue, like, because the ontology had been growing since, the early 2000. And there's some old practices that we had done to, either create terms like, try to grow rapidly, try to address, the needs that people had or might have in the future. So for example, there was projects to proactively introduce terms in case people need them in the future. But these terms, sometimes they ended up being used, but not always. So we had like a lot of unused terms. There was also the idea to have grouping terms, but at many different intermediate levels. And so sometimes it got so entangled that it was difficult to maintain, difficult to, find terms. So we're also removing some intermediate levels grouping terms that we believe are more confusing than helpful. I talked about the pre-composed terms already. There was also, and this is stuff that like the GoCam, way of thinking helped us really formalize. There was also many single step reactions in the biological aspect of the ontology. So for example, we had methylation and DNA methylation and phosphorylation, things like that. And so when you think about this, for like DNA methylation was a very good example because it's DNA methyltransferase activity. This is the function. But then there's DNA methylation in various kinds of pathways. So there's multiple ways that the DNA can get methylated and for multiple reasons, during development, also to shut down retrotransposon and so on. So when you try to translate this into a GoCam, you realize like it's not precise enough. And so we really try to more clearly separate function and process. There are also terms that refer to more than one ontological aspect. So sometimes there's like a nuclear something function or mitochondrial. And so again, we're trying to keep everything more separate. And there's also experimental assays and non-physiological substrates. Like, for example, there was rubidium transport. And this is used just because it's an analog of potassium. Most potassium transporters will transport rubidium, and it's easier to follow with radioactive markers. So, terms like this, when we realize this is just the experimental data and it's not representing the biology, we try to, remove these terms as much as possible. And so here I have examples of obsoleted terms. So phenotypes, process terms that should be represented like a BP, pre-composed terms. There's also subreaction and reaction mechanisms. So, like we don't capture the sulfur transfer from cysteine. This is no different from the actual enzymatic reaction. And there's also like very specific substrates that no enzyme has this specificity. So we now provide the specificity in the input in the model or in the annotation extensions. And so this has resulted in quite a decrease of the number of terms, especially in the process part of the ontology. There was also a decrease in the functions because we did a couple of years ago a cleanup of Go terms that are mapped to EC, Enzyme Commission. And so there was like a lot of terms that Enzyme Commission had obsoleted. We didn't have a process to maintain things up to date. Now we're doing a little bit better. So hopefully, if a term gets obsoleted at EC, now we are aware of it. So we try to go with external resources so that the experts that know about enzymes are the ones, kind of informing what kinds of reactions we should capture. Okay, and so I want to talk a little bit about our GoCam curation tool and Go curation tool. So we have a tool suite called Noctua. It's a web-based tool that is developed by Go Central to do standard Go annotation as well as GoCams. And so this is the welcome page, the landing page, we call it, when you get to Noctua. If you're logged in, you're allowed to create new models, either in the standard annotation editor or in the GoCam, we call it visual pathway editor. You can also see the existing models and edit them. And then there's a number of filters you can search by, in order to find models you're interested in. And so, like I hinted to in the previous slides, we have two main workbenches, one to do standard annotation and one to do GoCams. So the standard annotation editor is a very simple form. You enter your gene product, the relation, your Go term, the evidence, and then you can enter additional information. For the visual pathway editor, you can, like here, I'm just showing an existing pathway where you add a new annotation in that pathway. So we have this toolbox where you can add an activity unit or a protein complex in order to, if a protein complex catalyzes a reaction, you can use that box. Or if you want to put a chemical input-output, you use this box. And so the activity unit allows you to put the gene, its function, and then you can see the function is part of the process, and it's part of a component. And then here, using these little three dot more menus items, you can nest other BPs, for example, and add other evidence. So currently we have about 2,000 GoCam models in production because the models start in a status called development. People work on them when they're happy with them. Some groups even get other curators to review them. When they're happy with them, they become production models. This has about 10,000 different genes from 5 major contributing groups. So UniProt, who does a lot of human, but also some bacteria and other things. PUM-base, fish and yeast, fly-base for Drosophila, SGD for yeast, and MGI for mouse. So these are the groups that contribute the most models right now. Yeah, there's also, I didn't mention here, but there's also an ongoing project to convert all the Reactome models for human into GoCam models. And so these are not yet public, but this is going to add thousands of models once it's finished. So, and about this, SGD also had models that were based from MetaPsych. So they had the yeast psych metabolic pathways. And so these models were converted into GoCam, so we also have the ability to, we take the time to do conversion of existing other network tools, if these tools are not supporting the group anymore and they want to move into GoCam, we have been supporting that. So the GoCams can be downloaded, as like some kind of AL file with all the complexity. It's called turtle format. And there are also all the annotations in a GoCam. They're flattened down into a standard annotation. And whenever we can export the extensions. So it depends if the extensions are compliant or not with the standard annotation model. We also have a pathway widget, a pathway viewer widget. So this widget is available from the Go GitHub, and anybody can, embed it in their website. And then it shows models, like all the available models or whatever you select. So right now, Amigo has it on all its gene pages, Alliance of Genomes, UniProt, PUM-base, and fly-base. As far as I know, these are all the groups that have the pathway viewer widget. The GoCams are also available at a resource called Index. So this is an open source framework where, different pathway resources can connect together using like the same underlying data model. And so since a couple of years, they have, a couple of years, but it's getting better and better. We now have GoCam model available in here. We used to only have human, and now we have all the production models here. So they also have their own pathway viewer, where it shows very similar to what you see in the Go Central tools. And they also have this really neat feature, which is the iQuery feature, which allows to do some sort of, they call it a form of gene set enrichment analysis by clustering, finding, like you can input a set of genes and it's going to find the models that contain these set of genes and tell you which models are most enriched in your set. So this is an example using Index has a few test lists. So they have the death receptor list. And so here you see in red, this is part of the list. It stops at gas, but it's longer than that. This is about half the list. And then you can select, how you want to and like do the enrichment. So here I selected GoCam. And then when you run it, it finds which models are enriched. And when you look at each of the models, it tells you how many genes are found over the whole model. And then gives you a p-value for, your set of gene containing a significant number of those genes. And it highlights in the GoCam model which genes were in your query list. So that's very interesting because this is the kind of application we would really like to see. When we have more GoCams, it's going to become more significant. Yeah, so I see there's a question about the GoCam, so I'll answer that before moving on. Can you talk about the community usage reaction feedback on the better structure of the Go annotation? How do you collect this information? Yeah, well, so we don't have a formal way, I find, to interact with users. But what I can tell you about the reaction is like many groups come to us that are not historically part of the Go consortium, and they want to do GoCams. So there's there's several groups that contacted us. And so I think this is sign of some uptake. So that's kind of the right now, it's the best way that we have for new users is via GoCams, new contributors to the consortium. Yeah. So I hope that answers your question. Okay, and then I had like a couple of slides about how we use AI to enhance Go. So, everybody is in fact trying to make use of AI to, be more efficient. And so here's a little list of tasks that we do every day and that we would like to make more efficient with AI. So we, in ontology development, we create obsolete terms, but we also fix the hierarchy. Sometimes we're not sure of where a term should be. And we also have design patterns that, help make definition automatically. And it's been hard to do this manually. So hopefully at some point we can have more AI help doing this. There's also our tasks for annotation are to add new annotations, remove incorrect, do annotation reviews, sometimes on, like I said, specific topics. Also, we create new GoCams and we review GoCams and we do QC in all kinds of areas, and the ontology and the annotation and our mappings and all kinds of things. So any one of these, to have any help to be more efficient would be really excellent. So I'm just going to show a couple of slides of things that we started to play with. So Chris Mongol at Go, he's made quite a few little tools, but one of them is this gene annotation review browser. And so this is a annotation review, but it can do multiple things. It can suggest reviews, suggest new annotation, suggest deleting annotations or modifying annotations. So this is an example of a gene that is annotated to regulation of gene expression, and it's suggesting a more precise term, mRNA processing. And here you have the explanation, why it thinks you should review it. It's got references. Well, yeah, it's got the reference. So from the same paper, it suggested different annotation. And then likewise for the new annotations, you can have a gene and it's going to say, oh, you could annotate this to transcription regulator complex. And it explains why it should be annotated to that. And I think there's an extract of the text, so you can confirm with this. And in this case, yeah, it gives 2 references. So so this is something, yeah, we've been looking at. It suggests a lot of things, so now we need to see through which are the useful suggestions and which, how to prioritize such an amount of data for manual review, because right now they're not in a shape where they can just be taken in without reviewing. And then with respect to ontology editing, we also have agentic tools right now. So this is to explain how we do ontology editing right now. So we use this tool called Protege as the ontology editing tool. This is hooked to GitHub Actions for quality control checks. And so what we do is on the editor computer, there's the Protege loaded. And so you would, to create a new term, this is an example where you create a new term, you click on this little icon, new, you enter the name of the term, you click OK, then you have to open something to enter a definition, you click OK, then you have to click again to enter the namespace. So I didn't want to take screenshots of every step, but the steps are always like this. Add some more information, add a namespace, add a cross-reference. If you have multiple cross-references, you need to add them all in separate steps. You want to add synonyms and so on. So you do each, what they call annotation on the Go term one by one. Then when it's done, you save it. And then, with GitHub, we commit, we push the information to the GitHub on the internet, to our common version of the ontology, the master version. And over there, you create the pull request and you merge. So, it's quite a number of steps. And right now we have a different workflow, which is via an agentic workflow. So here is the same term, right? This happened just last week. The curator requests this term. She gives the RIA and the EC numbers, the definition, the PubMeds, and every information that we need. And so instead of going to Protege and entering everything one by one, the curator just asks our agent, who is called Dragon AI agent, please ask this term, please use this, and then add the cross-references. And magic happens, the term is there. So Dragon AI says, I have done what you asked me. But in addition, they have added some synonyms. And then they try to explain what they did. And so oftentimes when we do this, it's nice, it's fast, but we need to go and ask the agent to make some corrections because it just was a bit too creative and it did stuff that we didn't ask. But overall, I think that usually speeds up the process quite a bit. And so, yeah, this is it. Yeah, so this is just a slide that summarizes everything I said. I'll need to go over this, but just to conclude. And I put also a list of resources that you might find interesting if you want to, brush up on your Go skills. And yeah, this is a slide thanking the contributors. There's many different people, many different contributing groups. These are the active contributing groups. If I count every existing contributing group, it's at least twice or 3 times as much. So it's been a really nice, big, large consortium to work with.